This video is about the scientific concept of drug metabolism. For alternative medicine, see detoxification. Drug metabolism, also known as xenobiotic metabolism, is the biochemical modification of pharmaceutical substances, or xenobiotics respectively, by living organisms, usually through specialized enzymatic systems. Drug metabolism often converts lipophilic chemical compounds into more readily excreted hydrophilic products. The rate of metabolism determines the duration and intensity of a drug's pharmacological action. Xenobiotic metabolism is the set of metabolic pathways that modify the chemical structure of xenobiotics, which are compounds foreign to an organism's normal biochemistry, such as drugs and poisons. These pathways are a form of biotransformation present in all major groups of organisms, and are considered to be of ancient origin. These reactions often act to detoxify poisonous compounds. However, in some cases, the intermediates in xenobiotic metabolism can themselves be the cause of toxic effects. The reactions in these pathways are of particular interest in medicine as part of drug metabolism, and as a factor contributing to multi-drug resistance in infectious diseases and cancer chemotherapy. The actions of some drugs as substrates, or inhibitors of enzymes involved in xenobiotic metabolism, are a common reason for hazardous drug interactions. These pathways are also important in environmental science, with the xenobiotic metabolism of microorganisms determining whether a pollutant will be broken down during bioremediation or persist in the environment. The enzymes of xenobiotic metabolism, particularly the glutathione S-transferases, are also important in agriculture, since they may produce resistance to pesticides and herbicides. Drug metabolism is divided into three phases. In phase I, enzymes such as cytochrome P450 oxidases introduce reactive or polar groups into xenobiotics. These modified compounds are then conjugated to polar compounds in phase II reactions. These reactions are catalyzed by transferase enzymes such as glutathione S-transferases. Finally, in phase III, the conjugated xenobiotics may be further processed before being recognized by efflux transporters and pumped out of cells. The exact compounds an organism is exposed to will be largely unpredictable and may differ widely over time. These are major characteristics of xenobiotic toxic stress. The major challenge faced by xenobiotic detoxification systems is that they must be able to remove the almost limitless number of xenobiotic compounds from the complex mixture of chemicals involved in normal metabolism. The solution that has evolved to address this problem is an elegant combination of physical barriers and low-specificity enzymatic systems. All organisms use cell membranes as hydrophobic permeability barriers to control access to their internal environment. Polar compounds cannot diffuse across these cell membranes, and the uptake of useful molecules is mediated through transport proteins that specifically select substrates from the extracellular mixture. This selective uptake means that most hydrophilic molecules cannot enter cells, since they are not recognized by any specific transporters. In contrast, the diffusion of hydrophobic compounds across these barriers cannot be controlled, and organisms, therefore, cannot exclude lipid-soluble xenobiotics using membrane barriers. However, the existence of a permeability barrier means that organisms were able to evolve detoxification systems that exploit the hydrophobicity common to membrane-permeable xenobiotics. These systems therefore solve the specificity problem by possessing such broad substrate specificities that they metabolize almost any nonpolar compound. Useful metabolites are excluded, since they are polar, and in general, contain one or more charged groups. The detoxification of the reactive byproducts of normal metabolism cannot be achieved by the systems outlined above, because these species are derived from normal cellular constituents, and usually share their polar characteristics. However, since these compounds are few in number, Specific enzymes can recognize and remove them. Examples of these specific detoxification systems are the glyoxylase system, which removes the reactive aldehyde methylglyoxyl, and the various antioxidant systems that eliminate reactive oxygen species.